has about 25% damage. So that is real ancient human DNA. We can also show that that DNA is just from one person. And if we look at the Yersinia pestis DNA from the same specimen, we see about the same rate of damage, so about 25% damaged DNA. So that is old Yersinia pestis DNA. That cannot be just, let's say, 5 years, 10 years, or 100 years old. That is at least 150 years old. Likely, it is more than 500 years old. Because if we go back, we say 25% damage. We're somewhere here. So that would be between 500 and 2,000 years, which is kind of perfect. That's exactly where we want to see it. So it should be really old. So from this first study, we could then conclude, first of all, we have authentic human DNA, which is good. So there is DNA in those bones. But what we also have is authentic Yersinia pestis DNA. So there is pestis DNA in those bones. What we could also say then is that Yersinia pestis, so what causes bubonic plague, was actually one of the causative agents of the Black Death. Of course, we cannot exclude there was something else around, but certainly it was bubonic plague to some extent. But at that point, we don't want it to stop. We don't want it to stop by just saying, okay, it is really Yersinia pestis which has caused the Black Death. We wanted to go one step further. We wanted to see how did Yersinia pestis change in those last 660 years? How did it basically evolve? And that we wanted to do by targeting the entire genome, not just looking at one plasmid, but actually looking at the entire genome. And here we used a different approach because you can imagine long-range PCR products for the whole genome is a lot of work, and we don't want to do that. So instead what we used is actually fishing arrays, so capture arrays, microarrays that you can design. On one array, you can actually put the entire genome on the surface, which is quite handy because you can put 1 million probes, you can put a lot of them on the surface of the array, which is a little piece of glass, so you have all those probes on here. You incubate this array with your soup, and then the complementary fragments will bind. The background you can wash away, and then you can elute it and put it into the sequencing machine. So then you actually can capture the entire genome. And that actually worked rather well. So before array enrichment with shotgun sequencing, we had almost no Yersinia pestis DNA, and after enrichment, we had a really high proportion. So it was about 1 million-fold enrichment, which is really great and quite powerful. We were indeed able to put together from this enrichment almost the entire genome of those ancient bacteria from the medieval time. You can see here the coverage over the different plasmids is really high, except of PCP, because we didn't put it on the array because we had actually captured that before. So we didn't put that, and it also has a high copy number, so it would have probably also overswammed our sequencing. But what was really surprising then was when we then looked at the genome sequence, and one of the first things we did was making a phylogenetic tree, trying to find out how does the medieval plague compare to the modern plague. And what we actually found was that the Black Death strain, so our strain from the medieval time, is the most recent common ancestor of all modern human pathogenic strains. So if you want, it is the mother of modern pestis strains, which was a big surprise because I told you before, people had tried to date this divergence. They came up with like 8,000 years. But it's actually not 8,000 years, it's about 600 years. So actually the Black Death seems to have been the initial spread of plague in the world. That was basically the first big pandemic that has spread the plague in the world, where it still occurs in so many places today. What was also great is that we could actually use the fact that we know exactly how old the cemetery is. We know it's exactly 662 years. So we can use that to calibrate the molecular clock. So we have a dated fossil DNA now. And we can then also calculate, for example, earlier divergences, like the pathogen divergence to the human pathogenic strains, and that is less than 2,000 years. So plague itself as a disease is not more than 2,000 years old, which is also great because it kind of confirms those theories that most infectious diseases are rather young. So at least for plague, that is already true. What we could then also do is we could actually find out what has changed in those kind of last 2,000 years after it diverged from the rodent pathogen in the genome that might actually cause this bacteria to be human-specific. Because something seems to be different in the genome, that one thing is a rodent pathogen, but the other one can actually also infect humans. And if you look at the pure number, it's actually very low. It's just 12 mutations in at least genes that we know are important for virulence. So it's a very small number, and we are working now with functional people to actually exactly look at those genes, look at the proteins, and see what they actually do, and how could those mutations cause something to be a human pathogen or basically not being infectious to humans. But of course, this also causes one little problem. I told you at the beginning, all those big outbreaks of plagues, the Antonine plague, the Justinian plague, so earlier outbreaks of plague. However, they occurred much earlier, and they don't seem to be going back to any human pathogenic strain which is still around today. So that's kind of a problem, because that was nothing basically that's still around today goes back to this time period. Even though if it was a big pandemic, you would expect it should be still around. So that was quite interesting. And we didn't really know how that might be related, but I will come to that a bit later. So I just hurry up, I guess. So what can we then say from the genome-wide analysis about Yersinia pestis from the medieval time? First of all, we can say it's actually not so different from the modern strains, which was maybe one big surprise, because a lot of people said back in the medieval time it was a mass killer, but today it's much weaker. It's not so virulent anymore. But genetically, we can't say that, because it's the mother of them, and it doesn't have a single private substitution. It doesn't have a single position in the genome that you cannot find in one of the daughters, basically, one of the modern strains. So everything is present somewhere in the world. So it's actually not special, if you want. What you can then say also is, of course, the most recent common ancestor. So we have basically no reason right now to believe that the genetic makeup can explain why the medieval Yersinia pestis was so much more lethal, why it was so much more pathogenic than compared, for example, to the modern strains. One bit of more analysis, which is actually not published yet, just submitted, was that we then not only looked at the 20 genomes, but we actually looked at genotype data from about 300 Yersinia pestis strains from all over the world. So we looked about 1,000 SNP positions and put in our ancient genome as well. So our ancient genome is here again. This is branch 1 and branch 2. It's a bit of a different representation. So this is again the Black Death falling as a common ancestor, as the mother of the human pathogenic strains. Here is the rodent pathogenic strains, and if you now walk back to the rodent split down here, which occurred 2,000 years ago, you can actually see there is something in between. There's actually some cluster here of 11 sequences, which were isolated all from rodents, so not from people, but from marmots actually, that when you make this dating of the phylogenetic divergence times, it is a date of 700 to 900 years AD, so after Christ. And if we then go back here, we can actually see this would be something which would branch off exactly here in this time during the Justinian plague. So there still seem to be some strains out there in the world that actually go back to this time here. They're not very common, but they exist. So that actually probably confirms that the Justinian plague was also caused by bubonic plague. At least there was also some outbreak associated to this time period. But then, of course, we still would have, or would really like to explain the high mortality rate. So we would really like to explain why was it so much more lethal in the medieval time compared to the modern strains. Of course, there are many other factors that could be the case. We know the climate has changed. We had the little ice age occurring in the medieval time, so climatic changes could be responsible. We see differences in the vector population. Some people say it wasn't flea, it was actually lice. Some people say it was not the same rat, because we know the rat has changed. So the rat in the medieval time was Rattus rattus. Today we have Rattus norvegicus, so it's a different species. Rattus rattus is actually on the red list in Germany. It's almost extinct, which is quite amazing for a rat. So that has changed. And then, of course, it is also possible that we have changed, that basically we adapted. If 50% of the people die, the ones that are surviving are maybe more resistant to the disease. So the innate immune system, like HLA, for example, might have been better adapted, so the survivors are actually resistant against the next wave of a big pandemic. It could be that there were other diseases around, and of course the social conditions were quite different. It was much more unhygienic, and of course things have changed, fortunately, in the last 600 years. So what do we want to do then in the future? First of all, we want to look at earlier plagues, such as the Justinian plague, so basically use older skeletons. We want to look at more Black Death genomes. What we also want to do now is we want to actually look at human immunity-related genes. We want to study them in the population before the plague, during the plague, and after the plague to see if there are frequency changes. See if suddenly certain genes actually go up in frequency. And then basically try to identify them. We can even then predict to which protein in the pathogen they might actually bind. And we then also want to study more pathogens. We have already started with tuberculosis, leprosy, smallpox, syphilis, which are other mass killers. We are reconstructing their genome right now. So by that, I like to make a little bit of advertisement. I was just awarded an ERC grant, I have 2 postdoc positions, 2 PhD positions, so if I'm allowed to make a little bit of advertisement, I'm always looking for interested people. That's, I think, really quite exciting research because it's really a new field of research. Nobody's done anything like that before, and what we basically want to do is we want to provide a fossil record for pathogens, and I hope I could convince you how interesting that could be. So by that, I would like to thank, first of all, my 2 postdocs, and Olaf Ries, who gives me access to the labs in Tübingen, because I'm just a little junior professor right now, so it's hard sometimes to get your own lab as that, I can tell you. And then my 2 collaboration partners in Canada, as well as in Tübingen, and then people from the Max Planck Institute where I could use the sequencing facilities, and the Carl Zeiss Foundation, who's actually funding my position. So by that, thank you for your attention.